Alright, so in the last video we finished ethanol precipitation of large-scale PCR product. So now we have a concentrated volume of our DNA in order to run transcription, transcribing our DNA into RNA. For transcription, it's a 20 microliter reaction containing 3 microliters of water, 2 microliters of 10x amploscribed transcription buffer, 2 microliters of diethiothridol, 100 millimolar, 1.5 microliters of 100 millimolars of ATP, CTP, UTP, and GTP, 5 microliters of our double-strand DNA from LS-PCR, and 2 microliters of T7 RNA polymerase. So first we're going to add our water to the transcription reaction. 3 microliters. It's super important, especially in transcription, that you bring all of your reagents to room temperature and you vortex and spin them down, because GTP is prone to crash out in cold solution. And also because of that, you want to add GTP last. Okay, now we're going to add 2 microliters of the 10x amploscribed transcription buffer. Okay. Next we're going to add 2 microliters of diethiothridol, 100 millimolar. Okay, now we're going to add 1.5 microliters of 100 millimolar ATP. Next we're going to add 1.5 microliters of CTP. Next, 1.5 microliters of UTP. And then 1.5 microliters of your GTP. As I said before, if you don't properly bring your reagents to room temperature, GTP is prone to crash out. So I'm just going to check for any precipitate that is in the tube. And it looks like GTP did not crash out, so that's good. Okay, so now we're going to add 5 microliters of our dsDNA from large-scale PCR. Okay, now I'm going to get the T7 RNA polymerase from the freezer. Okay, we're going to add 2 microliters of T7 RNA polymerase to the transcription reaction. We're going to gently mix the reaction. Okay, now we've mixed up our transcription reaction. There's no precipitate in the bottom, which means GTP did not crash out. Now we're going to add the transcription reaction to the 37°C incubator overnight, around 18 hours. Here you can see I labeled my transcription reaction, put the date, my initials, aftermers, and please do not move. So yeah, this is going to go into the 37°C incubator overnight, around 18 hours, and then tomorrow we're going to start the second half of transcription, and after that we can run page. Okay, so yesterday we added the reagents to the transcription reaction. The reaction has incubated overnight at 37°C. Now we're going to add 1 microliter of DNase 1, mix gently with micropipette, and incubate again at 37°C for another 30 minutes. And this is to remove any DNA template left over in our RNA sample. So here I'm adding 1 microliter of DNase 1. Here's the transcription reaction. And I'm just going to mix gently. Okay, now we're going to incubate this at 37°C for 30 minutes. Okay, after the DNase has incubated with the transcription reaction for 30 minutes, we're going to add 21 microliters of 2X denaturing dye. The 2X denaturing dye contains urea, denaturing proteins, as well as EDTA, a chelating agent to remove any divalent cations, which is required for enzymes such as polymerase and DNase. It also has bromophenol blue. Bromophenol blue runs around 20 base pairs in 8% acrylamide gel. So in page, this is going to be our visualizing agent. The RNA shadow is going to be above the bromophenol blue, of course, because our RNA sample, our pool, N71 is around 112 base pairs, and the bromophenol blue runs around 20 base pairs in 8% acrylamide gel. So here I'm just adding the 2X denaturing dye. I am then going to heat denature the transcription reaction for 3 minutes at 65°C in the thermocycler, and this is going to be stable in the freezer until tomorrow when I run page. Tomorrow before I run page, I'm going to denature the transcription reaction again, and this is just to linearize our RNA and to get rid of any secondary structures so that the RNA can migrate through the gel uniformly.